Thank you for having me today. The topic I'm going to present is Unravel the Mysteries of STEAM. Invivogen is the first biotechnology company in France, which was founded in 1977 by Professor Chihabi, a microbiologist. In 1997, U.S. subsidiary was created in San Diego. Invivogen Asia was incorporated in 2014. Our bodies are constantly being exposed to infectious agents in evolutionary history. Now human beings are suffering from COVID-19 pandemic. However, we never give up to fight the diseases. Chinese began to practice a form of immunization by drying and inhaling powders derived from the crusts of smallpox lesions in year 1000. In 1798, Edward Jenner introduced a far safer method of deliberate infection with cowpox virus, which caused a mild infection that also induced immunity to human smallpox. By 1800, the procedure was referred to as vaccination, the part of adaptive immunity. The story of innate immunity should start with Mechnikov, who discovered phagocytes in 1882. However, scientists consider the phagocytosis as part of adaptive immunity until 1989. Charles Janeway raised the concept of innate immune system, which is non-specific and the first line of immune defense. T cells are activated by non-self-antigen, but not self-antigens. What is known by that time is T cell activation requires two signals. Signal 1 is antigen itself, that is presented by MHC to T cell receptor. Signal 2 relies on B7-mediated CD28 activation. But self-antigens are not sufficient to induce signal 2. Charlie hypothesized there are a set of receptors in antigen-presenting cells that directly recognize pathogens and control the expression of B7. Charlie proposed those receptors can recognize the common microbial structure, so he named them as pattern recognition receptors, short for PRRs. A graduate student named Ruslan Mezitov came across the literature. He was enchanted by the ideas and joined Charlie's lab as a postdoc fellow. Ruslan started to discover the PRs. Eventually, he successfully cloned Toll-like receptor and proved that can induce B7. After discovery of Toll-like receptor, a large collection of pathogen-associated molecular patterns are investigated. And many scientists contributed to discovering PRs. Professor Shizuo Akira particularly made a major role in the Toll-like receptor investigations. PRs localized on cell surface can trigger immune responses as soon as they encounter the microbes. However, when the microbes start replicating and injecting the nucleic acids into cytoplasm, there are cytosolic sensors waiting. In 2004, double-strand RNA sensor, RIGI, was discovered by Professor Fujita Takashi from Kyoto University. Since then, many cytosolic DNA sensors are illuminated, which majorly contributed to inflammasome and STING pathways. Since the discovery of STING, literatures in top journals are increasing year by year. The fundamental researches in immunology and oncology are profound. STING is short for stimulator of interferon genes, also known as TMEM173, ARIS, META, and MPRS. It was reported in 2008 by Glenn Barber's lab and Shuhong Bing's lab. Both labs described the downstream pathway as TBK1, IL-3, and eventually induced type 1 interferon. However, DNA and Sendai virus are not directly sensed by STING. What is cytosolic sensor that bridges STING pathway and what is the agonist of STING attracts massive studies since 2008. The breaking through was reported by Burdett in 2011. He he found cyclic GMP and cyclic AMP are STING agonists. Now the most famous cascades are cGAS STING that are discovered by James Chen's lab. In 2016, Daniel Stetson's lab tested the reported cytosolic DNA sensors by knockout scales. They conclude cGAS is the only cytosolic DNA sensor that triggers STING. Half years later, two publications in Nature Communication point out Fi16 was important for cGAS sensing DNA. Fi16 knockout impaired type I interferon induction. Fi16 was also important for STING sensing cGAP. The inconsistent results come from cell types. Here the author used macrophage. Daniel's lab used monocytes. Our in-house data showed consistent Fi16 phenotype when we differentiate THP1 into macrophages. The most studied pathway remains cGAS STING. Let me introduce how James Chen's lab discovered this pathway. They use DNA to stimulate a group of cells, then treat the cells with heating RNase and DNase. The cells with different treatment are subsequently applied in STING expression cells. However, they couldn't see the STING activation, so the protein, RNA, and DNA are excluded being as STING agonists. Eventually, cGAP is found by mass spectrometry. As cGAP is metabolized from ATP, there should be enzyme upstream. With the question, they find cyclic GMP AMP synthesis, short for cGAS, is the DNA sensor. Recently, there are some stories on cGAS localization. Jonathan Kagan reported cGAS localized on the plasma membrane. I'm looking forward to more discussions on the plasma membrane cGAS. I will not go through all the molecular mechanisms of cGAS STING. You may refer to the review by White-Hornack. It is the best summarized by far. I'm going to summarize the latest publications in 4 chapters: infectious diseases, cancer, autoimmunity, and adjuvant. The part of infectious disease include invasion, evasion, and interplay between different pathways. The DNA from Mycobacterial tuberculosis can activate cGAS-mediated STING response, in turn induce type I interferon production. At the same time, MTB DNA induced interleukin-1 beta secretion by activating AIM2 inflammasome pathway. As I mentioned in previous slide, cyclic AMP is the second messenger from bacteria. Here the author reported cyclic AMP from MTB can activate STING-mediated type I interferon induction. cGAS is the DNA sensor, but it does recognize RNA virus by different mechanisms. Polyglutamine binding protein 1 can capture the reverse transcript HIV DNA and interact with cGAS to initiate STING response. It was previously shown that NLRX1 functions as a negative regulator of innate immune response by inhibiting IL-1 and cytokine response. HIV is a very smart pathogen that can escape the immune response under this negative regulating system. However, the mechanism by which NLRX1 promotes HIV infection remains unexplored. Microbes are able to evade immune defense by directly interfering STING response. Tegument protein UL82 from human cytomegalovirus can interrupt STING response by blocking its trafficking from ER to Golgi apparatus. The protease NS2B3 from dengue virus can cleave human STING, but not murine STING. The following study reviews NS2B protease degrades cGAS during infection. Such degradation subsequently impairs type I interferon production in the infected cells. Poxin, a virus nucleus from poxvirus, can specifically degrade 2'3' cGAP. This is the first literature identifies intracellular nucleus that can cleave 2'3' cGAP. Let's go back to MTB. MTB phosphodiesterase, CDNP, can hydrolyze both cyclic AMP and host 2'3' cGAP. As a consequence, cGAS STING response is antagonized by MTB. I will not go through all the stories of aviation. Please find a nice summary in this review. A recent review summarized the innate defense against the coronavirus, including STING response. Please feel free to read. Interplay among different sensors is quite common in recognizing pathogens. During the early infection stage of malaria, cGAS STING activation induces SOCAS1, which limits the function of MyD88. As a result, TLR7 response is impaired. However, in the late infection stage, TLR7 remains the dominant defense against the malaria. Inflammasome is able to cross-regulate cGAS STING response after DNA virus infection. The author reveals that activated caspase-1 or caspase-4511 can cleave cGAS, which impairs the enzyme function of cGAS. Gasdermin D is key factor to induce pyroptosis, which is the hot topic of inflammasome. Francisella infection causes Gasdermin D cleavage. The Gasdermin D N-terminal forms Gasdermin D pole, which leads to potassium efflux. The author found potassium is important for cGAS enzyme function. You may see here the potassium removed culture medium significantly impaired cGAMP production. I'm pleased to introduce the pyroptosis and necroptosis reporter cell line, THP1 HMGB1 Lucia. Luciferase is fused with HMGB1. In the process of pyroptosis or necroptosis, HMGB1 is released. You may collect the supernatant applied in luciferase substrate. This is a good tool to monitor pyroptosis and necroptosis. Gram-positive bacteria can induce STING-mediated autophagy. You have to be careful as heat-killed bacteria will not induce autophagy. So the author emphasized VitaPEP in this study. We also provide tools for autophagy study. They are autophagy inducers, autophagy inhibitors, autophagy genes, and autophagy reporter cells. The second chapter is cancer research. How to convert the cold tumor to hot tumor and attract the self-immune cells, an eye-catchy topic in tumor immunotherapy. In this part, I will introduce the anti-tumor effect of STING from fundamental research to clinical trials. In 2011, Tom Gajewski's lab reported that tumor vaccine can induce type I interferon. They hypothesized innate immune sensors are involved. They tested TR4, TR9, RIG-I, and STING. Only STING knockout impaired the T-cell priming. The literature attracted global interest from both academia and pharmaceutical industry. Massive investments on tumor therapy are emerging since then. DMXAA is a small molecule STING ligand. It was a good candidate for lung cancer from Novartis. However, it was stopped in phase 3 in 1991. Now scientists start to accept the reason when the study reveals that human STING does not respond to DMXAA. Let's move forward. Adura Biotech discovered a CDN named ADOS100 and pushed to phase 1 clinical trial soon. they use our THP1 blue ISG cells as a STING agonist screen tool, which makes sure the drug candidate activates human STING. We can see they use many tests to prove the candidate is specific to STING recognition. However, after a dual publication, we released STING knockout reporter cells, which can make the specificity test much easier. I would like to introduce the reporter system from Invivogen. We have two reporter systems, secreted embryonic alkali phosphatase, short for SEEP, and secreted luciferase. After the stimulation with ligands, collect the supernatant and add the substrates of SEEP and luciferase. You may have the quantitative analysis by plate reader or luminometer. We provide human cell lines and murine cell lines for your STING research. Please note that THP1 endogenously harbors HAQ mutant. Therefore, we construct the wild-type STING knock-in THP1 reporter cells for your convenience. We also provide STING pathway knockout cells for your drug candidate screening and fundamental research. Let's go back to Adura's story. They test ADU-S100 in different tumor models and also melanoma-induced lung metastasis. The anti-tumor performance is profound. Novartis was attracted by the report and invested $750 million in 3 stages. The phase 1 data of ADU-S100 last year was not good. Novartis withdrew the remaining investment. Adura's stock dropped 30% soon. But the CEO of Adura announced that ADU-S100 is not dead. They planned phase 2 study in head and neck cancer. Let's hope they can have good results. I have summarized the clinical trials by far in this table. I won't go through all of them, but only pick up the one I believe very interesting. GSK published a small molecule STING agonist that shown a good anti-tumor effect by intravenous administration. It also activates several human STING variants and murine STING, which is good for animal study. Synlogic engineered E. coli niso to produce high levels of STING agonist, cyclidine AMP, which shown clear anti-tumor effect. You may also find the details from their latest publication in Nature Communication. Bioshape dose response is widely observed when naked CDN is administered. Intratumoral injection of naked CDN shows poor abscopal anti-tumor effect. Those defects are well solved by Kodiak by utilizing the exosome complexed CDN. Immune-stimulating antibody conjugate is a new trendy topic of immunotherapy. We have a dedicated project for antibody-drug conjugate with our CDN. Hopefully, we can release those products in 2020. Patents in CDN are very crowded. We start to focus STING quite early. Luckily, we can have a seat in this crowd. 164 CDNs are developed by Invivogen. Please read our publication for more details. I will pick up 2 CDNs from ours to compare with ADU-S100. All of them are commercially available. We tested a cytokine induction by candidate CDN. CL656 in green showed comparable or even better potency than ADU-S100 in blue. CL656 is much better than 2'3' cGAMP in black. We move on to test the anti-tumor effect. In CD26 colon tumor model, CL656 treatment significantly repressed the tumor growth and improved survival rate. The adaptive immune response induced by CL656 is also proved. In 80 days treated with CL656, we rechallenged the mass with CD26 color tumor. You can see the tumor growth is inhibited. AH1 tumor antigen is detectable after CL656 treated. We also tested the synergistic effect with immune checkpoint inhibitor. The combination of CL656 and anti-PD-1 significantly enhanced the survival rate. We changed the tumor model to melanoma. The anti-tumor effect of CL656 is also profound. If you are interested in our proprietary CDNs, please feel free to contact us. The last part of cancer, I want to spend some time introducing the latest fundamental research of anti-tumor. David Rolle's lab reported cGAS are constitutively activated in some types of tumor. They hypothesized that could be the marker for those tumors. HER2 can prevent STING-TBK1 association. As a consequence, STING-mediated anti-tumor immunity is impaired. This is an interesting study that reveals the interplay between checkpoint and STING pathway. David Rolle's lab challenged different tumor models with ADU-100 and discovered a CD8 T-cell independent but NK cell dependent anti-tumor fashion. Early in 2015, Yang Xin-Fu's lab reported an anti-tumor effect by blocking CD47, but this was STING dependent. The following study that was published early this year showed bifidobacteria oral administration with anti-CD47 can facilitate tumor regression. Those studies illuminate the important role of microbiota in anti-tumor. cGAS STING are crucial to human defense against the pathogens. However, aberrant activation of self-antigen has recently emerged as an underlying cause for autoinflammatory syndromes. The schematic cartoon summarized several mechanisms of autoimmune disorders. I'm going to discuss that in this chapter. Mutations in SAMHD cause severe congenital inflammatory disease, known as Aicardi-Gouttiere syndrome, short for AGS. The author reported that in SAMHD depleted cells, single-strand DNA are accumulated in cytosol, where they activate cGAS STING pathway to induce type I interferon. Mice liking DNase2 die during development because of the overproduction of type I interferon. However, DNase2 and F1 receptor double knockout mice are suffering from severe arthritis after 8 weeks. The authors report that DNase2 dependent polyarthritis is prevented by the loss of STING function. I will put two literatures here as they can make up a good story. TRAX1 is an exonuclease that digests the DNA. Loss of function mutations of TRAX1 are linked to Aicardi-Gouttiere syndrome. The authors of lower publication shows that genetic ablation of cGAS in TRAX1 knockout mice significantly reduce the inflammatory cytokines and rescue the mice. Even deletion just one allele of cGAS. Let's look at the upper literature. The F1 response to transfectant DNA in single allele of cGAS remains intact. Taken together, those data suggest new drugs block cGAS at dose-dependent manner might be efficacious on autoimmune disease and keep the immune response to the viral DNA. cGAS inhibitor RU521 was discovered in 2017. The author tested in AGS model as well. However, from our in-house test, the specificity is not good. I would suggest you to try the new released inhibitor, G140. It can specifically inhibit human cGAS in both IRF and NF-κB signaling. Recent reports show aspirin inactivates cGAS by promoting its acetylation. The acetylation of cGAS impaired its enzyme function. Back to AGS model. Aspirin can rescue TRAX1 null mice. SAVI is newly described pediatric symptom. This is totally STING-associated autoimmune disorders. The first report of SAVI was in 2014. The author discovered 3 STING variants that can constitutively induce interferon induction. We also construct the SAVI mutant knock-in reporter cells. You may also find the test of ligands and inhibitors here. H151 is the best STING inhibitor by far. Palmetylation of STING is essential for its assembly into multimeric complex, and in turn, recruiting downstream signaling factors. Blocking cysteine residue 91 of STING impairs the palmetylation. As a consequence, the downstream signaling pathway is inhibited. It works well for both human STING and murine STING, including SAVI mutants. H151 also blocks different CDNs, including ADO-S100. In addition to existing SAVI mutant, Glenn Barber's lab discovered a new STING variant that can cause acute inflammation. Now it is accepted as a new SAVI mutant. The ability to induce type I interferon is even stronger than the previous one. For your convenience, we also construct some cloning vectors that carry STING variants. They are commercially available. I'm pleased to introduce a nucleic acid transfection enhancer, NATE, which can significantly boost your transfection efficiencies in hard-to-transfect cell lines, such as THP1 and RAW cells. NATE has to be used with transfection reagents such as lipofectamine. You can see with NATE, the transfection efficiency is significantly enhanced in THP1 cells and RAW cells. We also provide the one-stop shop for selective antibiotics. Of note, Invivogen has the trademark of Ziosi. Biotech interest of STING blocking is increasing in recent years. You may find more information in this review. The last chapter is adjuvant. Classical adjuvant induce strong Th2 response with little or no Th1 response. The current challenge is to develop adjuvant which induce a strong Th1 response. PRR activation increase the host's ability to eliminate pathogens. Thus, the incorporation of PR ligands in vaccine formulation can improve and accelerate the induction of vaccine-specific response. A number of these agonists are now in clinical or late preclinical stage of vaccines. When combined PR ligands with alum or classical emulsion adjuvants, the immune response can be biased towards a Th1 response. cGAMP is demonstrated to have a good potential in influenza adjuvant. The author showed cGAMP can enhance the adaptive immune response and significantly enhance the anti-H5N1 antibody titer. 4 years later, the same lab used liposome complex cGAMP as the vaccination and successfully cross-protect different serotypes of influenza virus. The liposome complex cGAMP adjuvant is potential for universal influenza vaccines. This is a collaboration between Invivogen and the Paludan lab from Aarhus University. We tested with different CDNs. Interestingly, only one CDN showed well protection against HSV-2 exposure. This is a good candidate for HSV-2 vaccine. This is a publication from Aduro Biotech. They compared ADO-800 with tumor vaccine GM-CSF. ADO-800 with PD-1 blockade induced much better tumor regression. By far, we don't have GMP ligands, but we try to provide more quality control certificates on vaccine-grade ligands. We also provide commonly used adjuvant like AS03, L-hydrogel, FA, and CFA. In addition to 4 chapters, I'd like to summarize how cGAMP transfer among cells, which is a frequently asked question. cGAMP can be packaged in HIV particle when the virus buds up. During infection to other cells, cGAMP is released into cytoplasm and in turn activates STING signaling pathway. Cell membrane fusion after HIV infection can let the cGAMP spread intercellularly. Gap junction is modulated by connexins. It allows cGAMP to directly pass through the gate between cells. SLC19A1, a folate transporter, is also an importer for cGAMP, which localizes on plasma membrane. cGAMP can pass through P2X7 receptor, an ATP gate channel. VRAG is a chloride channel, but also allows cGAMP entering inside the cells. That are all the stories I want to talk about STING, but I believe they are not exhausted. cGAS has very important functions that are independent of STING pathway. Please refer to this review for more details. If you are interested in cGAS research, you won't miss the cGAS agonist from us. G3YSD, which is a better potency than VAC-V70, and it is specific to cGAS. Please find the validation data here and on Invivogen website. Invivogen is an expert in innate immunity, so we are very serious about the contamination of endotoxin. We use our dedicated reporter cells to test the endotoxin contamination from batch to batch. We are 100% sure our products are endotoxin-free, as the endotoxin detection method we use is more sensitive to LALA assay. This is a publication by US FDA. The authors tested endotoxin in commercial iPhone beta products and compared sensitivity between LALA assay from Lonza and Invivogen reporter cell lines. LALA assay reported no endotoxin contamination, while Invivogen reporter cell lines detected the impurities. In vivo experiments prove the presence of impurities, suggesting the reporter cell lines results are reliable. Before providing innate immunity-related products, Invivogen are famous for high-quality antimicrobial antibiotics. You may find our solutions to prevent or eliminate mycoplasma, bacteria, and fungi. I won't introduce all the anti-contamination products, but only 2. For prevention purpose, we are familiar with panstrep. However, panstrep has no effect on mycoplasma and fungi. You may consider adenomycin in your daily cell culture medium. It can prevent mycoplasma, bacteria, and fungi. Panstrep is also used to eliminate bacterial contamination. However, there are more and more panstrep-resistant bacterial strains. You may consider normal cure as a backup when you face the hard-to-treat contamination. Last but not least, we are gradually releasing the COVID-19 related research tools. Please visit Invivogen website for more details. Thank you very much for watching.